Okay. All right, so general announcements. I have graded your exam. I have them to hand back at the end of class today, as well as the lab exam for those of you who are in my lab. I have finished it, and just based on going through the material that I have to actually test you on, it's going to be very heavily ID based. So I just want that out there. There's going to be a lot of IDs. Today we are going to start on metabolism, kind of talking about really, why do we need it? What does it serve as far as a function for the purpose of life? So we're going to be going through that today, and then next week we're going to talk a little bit more about how it actually functions for us. So today's the why, next week we'll do the how. Before I get to that, just statistics of exam 3. We had several 100s, so congratulations on that. The average of the exam was a little lower than the last one. It was a 75, the median being a 78. Still within range of past exams, so I'm not particularly concerned. As a reminder, I do replace your lowest exam score with your highest exam score at the end of the semester, just to give you that little boost. But that will not happen until the very end of the semester when I have all of your exams in. So just keep that in mind. I am doing much better this semester about organizing your Blackboard page, so everything is going into the appropriate weighted section, so there won't be some surprise grade changes at the end. Also, the final exam schedule is out, so if you don't know this already, our final exam is going to be Monday, May 9th, 9:30 to 11:10 in this classroom. We have more time, but it's just a normal unit exam. It's not a cumulative exam, it's just the final unit, so it'll be the same length, same format as any of my other exams. Okay? All right, so I have been talking about energy throughout the last 2 semesters, kind of just been throwing it around, but we do actually have a definition for it. And as a heads up, we are going to get into some physics and a little bit of chemistry today. I know we've been avoiding that for a while, but we do have to circle back around to finish up this physiology portion. But energy can be defined as the capacity to cause change. So in the example of a human body, that's doing work. In order to move muscles, in order to turn things into proteins, we are changing the form of different materials, different molecules. And so we need energy in order to cause that change. Energy can exist in lots of different forms. There's potential, there's kinetic, there's chemical, there's heat energy. There's lots of different forms that energy can take. And these forms can then be used to do work. In the case of living organisms, in the case of a cell, most of the energy form that we're going to be using is the form of heat. That's usually what's associated with life, but there are other forms of energy. And the key takeaway here is that organisms need to get energy from outside our bodies. And there's a physics-based reason for this, and it's because of what we call the law, or the first law of thermodynamics. So the first law of thermodynamics is something of physics, so it is a law of physics. This is something that cannot be broken. And it says that energy of the universe is constant. It cannot be created, it cannot be destroyed. So the amount of energy that exists in the universe is finite. It's what we have, we're not destroying it, but we're not adding to it. We can, however, transfer energy or transform energy from different formats, and we can harness that to do things with that energy. But throughout that process, we're never losing energy, but we're not gaining energy either. So then comes in the second law of thermodynamics, and this one's the trickier one. I think the first one's pretty simple. We have what we have. Second one says that every time we transfer or transform energy, we are going to be increasing the entropy of the universe. In the case of organisms, again, usually that's in the form of releasing heat. Now, entropy, if you don't know what that is simply a measure of the order of the universe, and entropy is proceeding towards the disorderly direction. So what this law is stating is every time we transfer or transform energy, we are moving towards a more disorderly organization. We're going towards a lower energy state, is another way to put that. So another way to phrase that, or the outcome really, is that without the input of new energy, things can only become less orderly. That is the natural progression of the universe. Unless we are adding energy back in, it will always proceed in the direction of disorder, low energy. I have a couple of examples here. So this top one is a couple of fluids in a jar. So let's pretend that the yellow molecules are cold water and the blue ones are warm water. Okay? In this jar that we're starting with, they are ordered. They have stratified themselves because heat rises. The heated molecules are going to sit on top. The ones on bottom are going to be dropping. So they've stratified. Given time, just letting the energy flow as it does normally, letting the universe happen, they will become disordered. Those molecules will mix together. And so we started with something more orderly. We ended something with less order, less energy. Another example is a brick wall. Okay, we have a brick wall sitting in a field. Over time, that brick wall can only become less ordered. It can break down. It can start to crumble. On its own, that brick wall is not going to get any bigger. It's not going to get any stronger. It can only proceed in the direction of entropy. If I wanted to make that wall bigger, I can do that, but I have to add energy back into the system. I have to use energy to put bricks on that brick wall. So does everyone see how that functions? Just the general order. Okay. I know that this is a physics-heavy lecture, so if there's any questions, please let me know. There are other laws of thermodynamics, just as a heads up. I only care about these two because these are the only two that really have a direct influence on how life functions. So why does this matter? As humans, as living organisms, we have to spend energy constantly in order to fight entropy, in order to fight decay. In the case of a living organism, proceeding towards entropy is proceeding towards the decay of that organism. So we have to input energy in order to fight that. So another definition of life could be considered combating entropy in order to maintain homeostasis, in order to keep yourself functioning. And in order to do that, we have to spend energy. And as that law of thermodynamics states, we have to get that energy from somewhere. All right, so I've got a couple of examples here to help illustrate this particular concept. So here is a living person. They're sitting at their desk doing work, playing video games, I don't know, whatever they're doing. They are living, they are using energy, they are able to maintain homeostasis. However, if this person dies, they can no longer add energy to their system. So at this point, we cannot fight entropy. We're going to proceed towards entropy. We're going to proceed towards that decay. So as this person is sitting at their desk, they are currently in a state of high energy. They're sitting elevated, they're up above the floor. But prior to death, they had systems functioning, they had blood flowing through their body. As soon as we stop those functions, the blood is going to pool downwards. It's going to move to a level of lower energy. Eventually, the body decays. Those pieces start to fall apart. Again, we're not adding energy. We're simply proceeding towards a direction of low energy, more entropy. Those pieces will fall apart. They were sitting at high energy. They were sitting in that chair. Now they have fallen down. They have proceeded towards low energy system, and they are more disorderly. They are more entropy based. So that is the natural progression of a dead organism. It is going to go from something that was high energy, high functioning, requiring energy input. As soon as that energy input stops, everything falls apart. So here's another example. we're going to illustrate this example why that particular system separates us from things that are not alive, why life is different from non-life. And we're going to use me compared to a rock. So this was me, oh God, many years ago, prior to grad school, working in a quarry. I was looking for fossils. The rock and I are both considered high energy at this point in time. The rock is at a high altitude. I was up in a mountainous region. It was up high. I was in a quarry up at the top of the quarry, and so there was space for that rock to roll downhill. It is currently at a high energy altitude or high energy space. How did it get there? Well, that gets into geology, the volcanics and building of mountains. Those mountains lifted these rocks up. They now sit at a very high elevation, creating potential energy for this rock. You can also think of it as just like this precarious rock sitting on the top of a hillside. There is potential energy sitting there that could be used to send that rock down the hill. So it has high energy. It's also in a fairly large piece right now. Clearly that's not my goal. I'm about to break it open with a hammer and chisel, but before I did that, it's sitting in a fairly large piece. So we've got a large portion of rock. This statue is another example of that. A statue is a single piece. It has fallen down. It has broken into lots of pieces. It has proceeded towards entropy. That statue is not going to put itself back together. This rock, once I break it, is not going to put itself back together. Also, rocks contain high energy minerals. This again is geology based, but this rock right here, you see this green center surrounded by this kind of red crust. Those are high energy minerals that were created when these rocks formed in the base of a volcano, which is a very high energy, high temperature, high pressure situation. That's fine, those minerals are created in that situation, but as soon as it's erupted out of the volcano, those minerals start to decay because they're no longer in that high energy, high pressure situation. And they go from this, in this case, green mineral, and they decay towards this red mineral. So the rock I have has a bunch of high energy minerals that are going to proceed towards low energy minerals. They're going to decay over time. But now we can talk about why we're different. So we were both in high energy states. That we can agree on. However, the rock can only roll downhill. That rock is not going to go back up the hill on its own. It's going to roll downhill, and that's the only direction it can go. It can only get smaller. If I hit it with that hammer, it's not going to get any bigger. It's only going to proceed into smaller pieces. And it can only lose those high energy molecules. It can only decay those minerals into lower energy minerals. That is the only direction a rock can go. However, despite starting in the same position, I can also move uphill. I can go to a higher elevation. I can change my size to become larger. I don't want to, but I can. We can also make high energy molecules in our bodies from low energy. So we're going in an opposite direction. So this is what separates us from non-living things. We can reverse entropy. So a rock can only lose energy, lose altitude, lose its size, decay into low energy minerals. I can go down, but I can also go up. But the caveat is I can only do these things so long as I am actually alive and consuming energy. So in order to talk about that, we have to talk about what energy is actually in our systems. Whenever we're evaluating an energetic system, something that is using energy, we talk about its free energy. Free energy is just the energy in a system that's available to do work. So how much energy does this system have? Is it a rock at the top of the hill? Does it have high energy? Or is it a rock at the bottom of the hill? Does it have low energy? This example is a water wheel. We have a tank up here. It's very high in altitude, meaning it's very high in energy. That water can pour out of the high energy space. It can spin this wheel to do work, which will then power this little light bulb over here. But it only works because we had a lot of energy to start with. The same thing can be said for molecules. So here we have a nice big biomolecule. Each of these bonds represents energy. It is energy that is stored in that molecule. So if we break those bonds, we are able to harness that energy and use it for something else. So this big molecule has significantly more energy potential than this little molecule. This little molecule only has a couple of bonds that can be broken and taken energy from. And even if I had a whole bunch of little molecules, that is still not as much energy as these larger molecules. So we can actually estimate how much free energy is in any given system. And we do so by looking at how much energy is in our starting pieces, we call these the reactants, and how much energy is in the end pieces, the products. So every chemical reaction is going to start with reactants, that might be a biomolecule, and end with a product. We have a measure of energy at the beginning, we have a measure of energy at the end. The difference is the amount of energy that's available in that system. All right, I do want you to know the difference between these two energy pathways. The first we call the exergonic reaction. And this is a system where the product, the end piece, has less energy than the reactant. So we start with high energy, we end with low energy. The second is the endergonic reaction, and it's exactly the opposite. So our reactants have low energy, our products have high energy. So we're starting with less and ending with more. So if we look at each of these individually, the exergonic reaction starts with lots of energy, ends with less energy. This process, since we start with a lot and end with little, means that energy has to have gone somewhere. It's going to be releasing energy as part of that reaction. So we're getting rid of energy within that system. That means that an exergonic reaction is proceeding towards entropy. It is proceeding towards that disorder. It is proceeding towards a direction of lower energy. However, that also means that it can occur spontaneously. This reaction doesn't need energy in order to happen. It already has more than it needs because the starting point has more energy than the end point. So this kind of reaction happens on its own. One way I remember these two, exergonic sounds like exit. The energy is exiting the system. The endergonic reaction starts with less and ends with more. That can't just happen. We have to add energy to that system for that reaction to occur. So an endergonic reaction requires the input of energy from some outside source. So in this case, we're actually reversing entropy. We are creating more order. We are going from a low energy state to a high energy state. So again, you can see we have the input of energy here. In order for this reaction to happen, you have to have that input of energy. So this reaction cannot happen spontaneously. It needs that external source of energy or it will not operate. So again, the way I remember these two, endergonic has an N. It needs energy in order to operate. Okay. It is the endergonic reactions that allow us to live. It allows us to fight decay, to defy that entropy progression that is the natural progression of the universe. If everything wants to be that spontaneous release of energy, the exergonic reactions, in order for us to maintain our homeostasis, we have to have a constant input of energy in order to create those higher energy states. So this is what allows us to resist turning into a skeleton as long as we are still alive and consuming energy. So that input, that needed energy, is our consumption of food. All right, so how does that actually work? That's where metabolism comes into play. So now we've talked about the physics behind why we need it. Now we can talk a little bit about what it actually does. How is it operating as far as a physics standpoint goes? So the metabolism is the totality of an organism's chemical reactions. All of the reactions your body is doing in order to move your muscles, to create proteins, to duplicate DNA and reproduce cells, all of that is part of your metabolism. It's what's keeping us alive, what is allowing us to defy that entropy. In order for the metabolism to function, we have to obtain energy to allow us to cause that energy change. Your metabolism operates in different types of pathways. Any pathway, we'll get to the specifics, but any metabolic pathway begins with a specific molecule, that would be your reactant in those reactions, and a product, so the end point. However, metabolic pathways are not A to B. They're usually A to B to C to D to F. There's multiple steps along a singular metabolic pathway. In order for this reaction to go from our reactant to our product, we need some help. And that's where enzymes come in. So I've been throwing that around too. Now we can actually talk about them. Enzymes are going to be what facilitate each of these steps. And each step, each little piece of this metabolic reaction is going to require a specific enzyme. In particular, we are most interested in catalysts. So catalysts are a chemical that are going to facilitate a reaction, but they are not consumed by that reaction. And in fact, they actually speed up the reactions. So they allow these reactions to happen more easily. And that's what a majority of your enzymes are. They are catalysts. So they are a specific type of protein that is going to facilitate the function of change, and they're going to help those reactions happen faster than they would on their own. So in this example, we've got two different reactants. We want to turn them into this product. In order to do so, we add this catalytic enzyme, this protein, which is going to not only facilitate, it's going to allow this to happen, but it's going to help this reaction happen with less energy. So on its own, going from these two to this one takes a lot of energy. If we add the catalyst, less energy is required to do that job. So it just facilitates, it helps it happen easier. And then as you notice with this equation up here, not only do we get the product we want, but that catalyst is not used. So the catalyst remains and can be used again to facilitate that reaction over again. Okay. So the enzymes are going to be interacting with our reactants. Now, because we're biology and we like to rename things, we call these substrates. This is the reactant in that reactant-product reaction. So we just call them substrates in this case when they're interacting specifically with an enzyme. That little substrate comes along, and we want to change it somehow. We want to proceed along our metabolic pathway. In order to do that, it will bind to the active site right here of our enzyme. And once it's bound to it, once it forms a molecular bond, it becomes what we call the enzyme substrate complex, a very creative name. So they have bound together now. We have a single larger molecule that is formed from a enzyme protein and our substrate. While they are bound together, the enzyme is breaking molecule bonds and reforming them to change that substrate into a final product. Okay. So here's another figure just showing the same thing. Here we have two substrates, two reactants, and we want to get two different products. Those substrates come in, they bind to the active site of our enzyme, forming our enzyme substrate complex. It forms different bonds, it changes the chemical structure of these two, and that enzyme can then release those products, which are now a new form, which are either going to be the end of our metabolic pathway or simply the next step before the next change that needs to happen. That enzyme is not used up. Remember, the catalysts keep going. So that enzyme is now ready to bind to the next set of reactants or substrate. So it's a cycle. It binds, it changes, it releases, binds, changes, releases. So I've been talking about them as, you have your starting point of energy and your end point of energy. Chemical reactions in a living organism are a little bit more complicated. We do actually have to add a tiny bit of energy at the beginning in order for those reactions to happen. So in the example of an exergonic reaction, the one that should happen spontaneously, right? We start with high energy and we're going to low energy. High energy to low energy. I said that happens spontaneously. It will happen on its own, which is still true, but we do have to add a little bit of energy to get it over what we call the activation energy. So activation energy is the amount of energy that this reaction needs to spend in order to get to a point where it can happen spontaneously. This is not going to be as much of a problem with our endergonic reactions. They still have an activation energy, but we're already going towards a higher energy state. So in this case, this is our normal reaction. We have to get over this activation energy bump. And once we reach that peak, it will happen on its own because we're going towards a state of lower energy. Enzymes help us by lowering that activation energy. They're lowering the amount of energy that this cell needs to spend in order to get this reaction to happen on its own. So rather than spending this much energy, the enzyme allows us to spend this much energy, and then it will happen. Another way to think about this, just another way to illustrate this, if we want to roll a rock downhill, we just have to get it over this little hill that's in the way. So we spend a little bit of energy to get it over this hill. Once it reaches the top of that hill, it will roll down just fine. That process will happen. So we just have to put in a little bit of energy to get it over that hump. And the enzymes make that hill just a little bit shorter. Oh, same picture. Now there's something else that we've talked about that helped this process. I realized after I had given Wednesday's lecture that I called these something else. The names are equivalent, so it doesn't matter to me which one you want to use. Coenzymes are something I talked about on Wednesday. They are also called cofactors. I don't, again, care which one you want to use, they're fine. But these are non-protein enzyme helpers. They are going to activate those enzymes to tell the enzyme to do that substrate binding complex. And most of our coenzymes are vitamins. So this is why vitamins are so essential to the functioning of the human body, because they are these coenzymes that are going to bind with enzymes and help those enzymes do their job, help them change a reactant into a product. So here we have an example. Here's our little enzyme. Here's the reactant, the substance that we want to change. It can bind to my enzyme, but it's not complete. It's not going to actually change that reactant into a product unless my coenzyme, my cofactor from my vitamin, comes in and docks with my enzyme first, and that completes the activation site. Now the substrate can bind, the enzyme can change its molecular structure, and we can proceed along the metabolic pathway. So we need these vitamins so that we have the coenzymes we need to activate enzymes so that they can do their job. Now, these are ones that are helping enzymes. There are also things within our body that are going to be inhibiting enzyme function. Some of these are going to be natural. They're part of our natural metabolic monitoring, making sure we're not making too much or too little of something. But these can also be things like toxins, pathogens, things that are going to bind to our enzymes and prevent them from doing their job. And we call these inhibitors. So inhibitors are going to shut down the operation of an enzyme. I'm focusing on the ones that are specifically functioning for our body. They're normal part of our function. But like I said, toxins can be inhibitors that are going to block the function of these enzymes. There are two categories. We have competitive inhibitors and non-competitive inhibitors. Competitive inhibitors are going to bind to the active site of the enzyme. So where that substrate, where that reactant would be binding in order to be changed. Because it is binding in the active site, our substrate can't bind anymore. There's no room for it. So it's physically blocking it. That's why we say it's competitive, because it's competing for that active site. Because of that, this reaction won't take place because we don't have a substrate to change. The non-competitive inhibitors bind to a completely different space on the enzyme. So the back side of the enzyme, for example. In this case, the active site is still there, so the substrate could bind there. However, when a non-competitive inhibitor binds to an enzyme, it starts to change its shape. And it can have a couple of different outcomes. Either it changes it so much that that reactant can no longer bind there, it's not the right shape anymore. Or it changes it just enough that it can still bind, but it's not going to be nearly as efficient. So you're not going to quite get as much product out of this reaction. Okay. Now, our metabolic pathways can actually use this inhibition factor in order to, again, monitor our outcomes. So maintain the level of particular products that we want within our system to make sure that we're not overproducing. And we call this feedback inhibition. So in this case, the end product of the metabolic pathway, so it's gone through its steps, it's created its end product. That end product, if we have enough of it, the body says we don't need any more right now, will actually bind back to that first enzyme and block the active site. So it's its own self-regulation. This is done so that the cell is not wasting any of its materials. We don't want to make too much of a product if we don't have a need for it. So it's going to make sure that we maintain the correct levels without wasting anything. I'm zooming in just so you can see this a little bit better. So here's our metabolic pathway. We have our first enzyme. It binds with a substrate. It goes through a multi-step process using multiple enzymes along the way. Each of these enzymes are different, they're specific, and it creates a product. That product is being used by the cell, but if we reach a level where we have enough, we don't need any more right now, that end product is going to come back around and bind into the active site of our first enzyme, effectively blocking that metabolic pathway. So this reaction can no longer continue until the cell says, I need more of that product. That product will be expelled from the enzyme, and this process can happen again. Okay. There are two main directions that metabolic pathways can take. The first is catabolic pathway, and this is one where we are releasing energy by breaking down larger, more complex molecules and creating smaller, less complex molecules. So it makes sense. This is an exergonic reaction. We're starting with something high energy, a large biomolecule, and we're going to something with lower energy, a smaller biomolecule. This example is showing you sucrose, which is two bonded pieces of glucose. This is a larger molecule, not a particularly complex one, but it is something our body does. It binds to its enzyme. That enzyme breaks that bond. It releases energy. And our product are two smaller glucose molecules. So this is our process of breaking down food for digestion. This is an exergonic reaction. We take larger biomolecules provided by the food we eat, we break it down, and we release the energy in those bonds. Okay? Everyone make that connection? Good. I know this is not the most exciting lecture, but it is a very essential part of how we function. The second metabolic pathway is the anabolic pathway. So these are going to be the reverse. We need to consume energy for this reaction to make larger, more complex molecules from smaller, less complex molecules. So again, here's my example. We have two little glucoses. They're all by themselves. They're smaller, less complex. They bind to an enzyme. That enzyme takes energy in, reforms that bond, and creates a sucrose molecule, something that is larger and more complex. So an anabolic pathway is an example of an endergonic reaction. We're going from low energy to high energy, and we have to take in energy to do it. So a good example of this would be the creation of proteins. If you remember way back when we talked about that, proteins are made up of amino acids that are strung together. Amino acids are smaller, less energy pieces, but as soon as we start stringing them together, we create these very complex structures that fold in on themselves to create a functioning protein. And proteins do all of the work for our bodies. So this very important reaction of creating proteins is an endergonic reaction that requires energy input. One way I remember this versus catabolic is that anabolic and endergonic both have ends. You might also think of anabolic steroids. Anabolic steroids are things that are going to increase muscle mass. They're creating something larger. That's the same direction that anabolic pathways are taking. They're going from a smaller thing to a larger thing. However, you can get these straight in your head. All right. Now all I have been talking about is going to circle back down to a very important molecule. This is called ATP. You've heard it before. I know I've thrown it around. We're going to start talking about how it's actually made now. This is called adenosine triphosphate. You should be aware of this word because ATP is such an important molecule for the function of a human. It is the cell's energy shuttle. This is how we can take energy from one reaction and give it to another reaction. We can transport it around a cell to facilitate all the different functions that that cell needs to do that require energy. So it's carrying energy and it's moving around the cell to give that energy to the reactions that need it. So a cell spends ATP, this currency, in order to do its reactions, in order to do the work it needs to function. So it can also be used to drive those endergonic reactions that we need to create these larger proteins, for example. So in this figure, we have a protein pump. We're trying to create a membrane potential. We're trying to create a electrical imbalance here. In order to send this little hydrogen proton against its gradient, we need to input energy. And it comes in the form of ATP. It gets shuttled from somewhere else in the cell. That ATP gives off some of its energy. This proton pump harnesses that energy to send that hydrogen outside the cell. ATP is built from a ribose, a sugar. That's what this green part is right here. I do want you to know the general shape, function of, or structure of ATP. So I'm not going to ask you to draw this by any means, but to have an idea that you have a central sugar that on one end has an adenine, which is a nitrogenous base, and on the other end, and this is the important end, it has 3 phosphate groups. These 3 phosphate groups are surrounded by oxygens. You may recall that oxygen is extremely reactive, meaning There is a lot of energy stored in these 3 phosphates. So when we need to use ATP to give energy to a system, what's happening is it bonds to that molecule and it loses a phosphate. One of these phosphate detaches. So we're breaking a bond and it's releasing energy from that bond that can then be used. The final molecule, once it's lost that phosphate, has become ADP, adenine diphosphate. So now it has di 2 rather than the tri 3. Now it's important to keep in mind that that phosphate that's been broken off is not lost. All we did was break the bond because we wanted the energy in that bond. So those phosphates are still floating around, meaning we can reform ATP from ADP. All we have to do is reform that bond. Here's another example. This is, again, a proton pump doing something. It needs to shuttle something against its gradient, so ATP comes in, it binds with the molecule, it breaks off a phosphate, so here's my extra phosphate that gets sent away. This protein is able to do its job because it harnessed the energy from that bond, and it releases an ADP and that little lonely phosphate. Another example, we'll skip over that. I want to make sure we finish. So ATP can be renewed, like I said. So we have the ADPs floating around and we have loose phosphates. So all we need to do is reform that bond. In order to do that, we spend energy. So it is an endergonic reaction. So taking our ADP, a smaller molecule, we add energy to it in order to make it a larger molecule, ATP. So this is why we eat food. We break down food to get energy so that we can reform ATP. So we use an exergonic reaction, the process of breaking down food molecules to get the energy from their bonds, to power this all-important endergonic reaction, the remaking of ATP, which is the energy currency of our cells. So can you make that connection? That we have to break down food, an exergonic reaction, in order to reform the energy we need for our cells. Okay? So it's not actually the food or the energy that we directly get from our food molecules that we're using. We're using that energy to drive a different energy creation reaction. So we can summarize this process in essentially 4 big steps. We consume food, large, high-energy organic molecules. We break down that food in exergonic catabolic reactions. You should make these connections, that exergonic, catabolic go together. That process is breaking down large molecules into smaller molecules, which are giving off energy. We take that energy from breaking those molecules, and we use that to power the creation of ATP, which is an endergonic anabolic reaction. It needs energy to function. We get that energy from breaking down food. And then we spend that energy, that ATP, to power other endergonic reactions around the cell, other things like building proteins that need energy input in order to happen. Okay? So you could really summarize how does life function in these 4 steps. Okay? And all of that comes down to the sole source of energy in this particular solar system, and that's from the sun. Now, like I said, first law of thermodynamics, we're not creating energy. The sun is giving off energy from the breakdown of particular chemical reactions within its core. That cannot operate forever. Eventually the sun will decay and it will break down. But that's a very slow and a very long process, so we're good for a while. But it is giving off energy that is being harnessed by organisms on this planet, in this case plants. They do photosynthesis, which captures that energy from sunlight. They turn it into larger biomolecules, and then we eat those plants, or other organisms eat those plants, and we eat those organisms, and we take energy from that. So all of the energy that allows you to function directly comes from the sun's process. All right, so key topics. Those 2 laws of thermodynamics, remember there are others. I only care about the first and second. I really want you to get, why is energy consumption necessary for life? Why is that needed for us to function? The terminology about metabolism, so the types of reactions and those different pathways that I went through, exergonic, endergonic, catabolic, anabolic, all of that. How do enzymes operate? What things help them? What things help or hurt them? And then ATP structure and the generalities of how it's created and how it's broken apart. We'll talk about how it's used next week. All right, do I have time for the exams? A couple minutes.